Good afternoon. For the providers joining today, my name is Mabel Gutierrez. I'm one of the pediatric infectious disease specialists at TMC. Thank you for joining to this session where we're going to talk about issues associated with COVID-19 vaccines. And so let me share the presentation for you. Okay, can you see?
Yes, thank you.
Okay. If you have any problems with the audio, just let me know.
Thank you.
So as we know that there's no specific treatment for COVID-19, there is no vaccine available so far. And on this end there's an urgency for vaccine development. And on this end, it's very important to set up expectations up front for the community to know what these potential vaccines might be able to achieve and what not. Because at the end, if at least one of these vaccines complete these randomized controlled trials, they wouldn't do any good if at the end they keep up being rejected by a population or by the public that is confused and that is disappointed by a vaccine or by anything less than complete protection secondary to this vaccine. So along this presentation, you're going to be able to gather some information, and at the end, in terms of vaccine development in relationship to SARS CoV2, but particularly, what is what we need in a vaccine candidate? So we need a vaccine candidate that is able to mirror or to surpass the natural protective immunity secondary to SARS CoV2. How many vaccines platforms there are currently in the current pipeline? So we're going to review several of those. Why this process take that long? So we're gonna familiarize with the standardized process for vaccine development. And also there's so many issues associated with vaccine safety. And with this information that I'm gonna provide, at the end, you are gonna be the judge about what to believe or not. Because all these vaccines, at least the four these vaccines projects from these vaccines pharmaceutical labs, I mean, all of those claim to be safe, immunogenic, very well tolerated. So you're going to be able to decide about this with a brief analysis that we're going to do. And we're talking about the vaccines from Oxford University, Cansino, Moderna, and Sinovac. So first, we are going to have to talk about the structural components. First, as you know, you're very well familiar with the S protein. This is the structural proteins or in the proteins that are in the surface of SARS CoV2. And then you can identify the envelope protein, the matrix, the nucleocapsid that case the RNA, and particular attention has been centered on. Can you hear me?
Well? I can hear you well. Yes.
Okay, there is some noise in the background, so I don't know. Can you mute yourself?
Sure.
I don't know if it's coming from it, but if it's not.
Can you hear the noise again? Maybe is the fan here. I can turn it off.
Oh, yeah.
Would that be better?
Would you? So, stop sharing.
Should I turn it off?
1 second. Okay, I think that is better. Can you hear me? Okay. So now we have particular attention has been placed to the S protein. This S protein has, we can see here the three-dimensional structure because that is very important because there's different settings where this S protein has to be coded from the genome. And this makes a different antigen and quality of the antigen. And there is a particular part in this S protein that there's lots of attention about, that is a receptor binding domain, that is this particular part that binds or is linked into the ACE2 receptor to allow the entrance into the cells. So, and this is important because different vaccine targets, they have different antigens. Some vaccine candidates may use the whole virus. Other ones may use a particular proteins like the spike proteins. Others might use the RNA, the ADN that code the information from this spike protein. And in these following slides, so we can see that this is the schematic representation of the genome of SARS CoV2. And this is the genetic coding for the structural proteins, particular attention to the S protein. And the S protein has multiple factors, and we are going to focus on one particular one that gets lots of attention, that is the receptor binding domain, as you can see here. Overall, the S protein has different structures or different components, and different vaccines has played with different formulations of the S proteins to be presented as their main antigen. So, but what is what we're looking for in an adequate vaccine candidate? So we're looking for a vaccine that is able to imitate natural immune response, at least to be the same or a little bit better or a lot better. So, and the natural immune response, once SARS CoV2 enters into the organism, is composed of two arms. The first one is the innate immune response, and the other one is the adaptive immune response. The innate immune response is, as you can read, is the first line defense against the viral infection and the entry site. And in this graph, you can see the virus that is interacting with the epithelial cells of the respiratory area. We have endothelial cells and the alveolar macrophages. Secondary to this interaction, we have pro-inflammatory cytokines, IL-6, macrophage, inflammatory proteins, and these cytokines overall attract myeloid cells, T cells, monocytes, macrophages, and particularly T cells are going to secrete interferon gamma, that is the main effector of this innate immune response. Subsequently, this will promote more inflammation and generates a pro-inflammatory feedback loop. Under healthy circumstances, this response has the final goal of suppress viral replication and dissemination at the earliest stage. Just a very balanced and measured response at the level of the respiratory epithelium. But then several weeks after that, we have the adaptive immune response. Usually it takes 1 week or 2 weeks to have some response. And usually we prefer the adaptive immune response because it's more specific and because it has memory. So we can follow the numbers here. Initially, the adaptive immune response is compounded by the cellular immunity. So we have the recruitment of SARS CoV2 specific T cells into the respiratory epithelium. We have the TCD8s that recognize, eliminates infected cells. Then we have the TCD4s that will prompt or prime B cells with the formation of neutralizing antibodies. And the neutralizing antibodies are this particular set of antibodies that are not just binding antibodies. Neutralizing antibodies are the ones that bind a particular area in the S protein, that is the RBD, and will avoid the virus to enter into the cells. Here in the graphic number 2, you can see the neutralizing antibodies that is binding and inactivating the virus so it cannot enter into the cells. And once we have the complex of the neutralizing antibody with the virus, so the alveolar macrophages will eliminate those as well as the cells that are infected by the virus. So this adaptive immune response at the end that is composed by B cells and T cells, when it's, let's say, balanced, measured, has a final goal of eliminate the virus and the cells that are infected by the virus with no further spread into other organs, just limited into the respiratory epithelium without producing further tissue injury. But then let's look a little bit deep into what is the role of the B cells and T cells in controlling COVID-19. So we have the B cell response that usually shows up about 1 week or 2 weeks after the onset of the symptoms. And we have initially antibodies secondary to the N protein, and after that, antibodies against the S protein. We have in this group binding antibodies, and a particular group of those they're called neutralizing antibodies that are to the S proteins that are the ones that we were just talking about. And if you remember, over the past couple of months, there's different articles that show that patients that recover from COVID-19, they have high titers of neutralizing antibodies. In that case, we belong to this group in which neutralizing antibodies are present, but also there's these 30% of patients that after recovering for COVID-19 will have no neutralizing antibodies. So the main question is, what is a part of the immune response that is producing immunity in this set of patients and is regularly assumed that is the T cell response. And the T cell response is usually starts 1 week or 2 weeks after the starting of the symptoms. We have two components in the T cells. We have the TCD8s that, as you remember, they are eliminating virus infected by cells. And then we have the TCD4s that has multiple functions, primes B cells to form neutralizing antibodies, is inducing TCD8s to eliminate viruses and the virus infected cells. And then overall, TCD4 cells trigger a cascade of inflammatory cytokines that is very well known and studied for SARS coronavirus. It's not 100% defined for SARS CoV2, but in the case of SARS coronavirus, produce specific cytokines, that is interferon gamma, TNF, IL-2, and these produce recruitment of immune cells, monocytes and T cells to the site that is the infecting site. And for that reason, we see lymphopenia in patients with SARS coronavirus. And overall, this brings a response that is called Th1 response to control the infection. This response is measured, is balanced. The only objective is to eliminate the virus and cells that are infected by the virus with no further tissue injury. Overall, after this response is triggered, we have the SARS coronavirus specific memory T cells that has been demonstrated that may last up to 2 years after the infection, after the patient has been infected. And overall, we have memory secondary to this T cell response based on TCD8 effector response and the TCD4 response. But then what is in a vaccine candidate, what is the type of cellular response that we're looking for? Because we need to know that in order to see what vaccine might be more convenient in terms of avoiding side effects. So we have several types of cellular response, but the ones that are more concerning and pertinent for this type of vaccines is the Th1 cell response, that are the main cellular response that provides cellular immunity to control the infection. And traditionally, these responses are known because they kill. It's the typical model of the intracellular parasites. The main effector of this response is interferon gamma, that is the main Th1 cytokine that it has been demonstrated that is associated with natural infections secondary to SARS CoV2. So overall, Th1 cell response is a response that has as the objective to produce some degree of inflammation that is measured, that is balanced in order to eliminate the virus and the infected cells that are infected by the virus. But sometimes the Th1 cell response might get out of control. And in that particular case, we have the Th2 response that might keep it in check. And then, there are some circumstances where the Th2 cell response might trigger an inflammatory cascade that produces tissue damage and is not protective, but otherwise is producing tissue injury or damage. And in particular, in the settings of vaccines, adverse events are associated with what we call vaccine enhanced disease. So under some circumstances, there is once SARS-CoV-2 has been infected the individual, so there is a defective T cell response that trigger allergic inflammation with antibodies that are not completely functional, and these, instead of producing a neutralizing effect, are inducing formation of immune complexes complement eosinophilic infiltration. So the typical Th2 response is the one that is associated with airway hypersensitivity or allergic reactions. There are some cytokines that are very well recognized to be associated with this type of Th2 cell response, that is the IL-4, IL-5, IL-13, IL-10. So once we have a tissue in which these interleukins are identified, so we have some knowledge that probably is a Th2 type response that is happening in that tissue. So overall, when we have vaccines, we have gene-based vaccines and protein-based vaccines. The current knowledge is that gene-based vaccines as DNA or RNA mainly induces a CD8 T cell response that subsequently will involve CD4s, and mainly the main response that will trigger is a Th1 type response. So overall, for the purposes of vaccines, that the adequate vaccine candidate for SARS CoV2, there's conflicting data in regards to if we want a type of Th2 cell response or Th17, because both are linked to this type of reactions or immune dysregulations with severe inflammations to produce tissue injury. How do we know that the adequate or the ideal vaccine candidate is able to produce protection in individuals that are going to be exposed or going to have natural exposure to the virus? So what we can measure in order to know if this virus actually the vaccine has given some protection to the subjects. So there's different things that we can measure. We can measure the humoral response that is linked to the B cells, and on that end, we can measure neutralizing antibodies, but these neutralizing antibodies are measured through the plaque reduction neutralization test. Neutralizing antibodies are not measured through the type of IgG serology testing that you can get in commercial labs. It's a specific testing that is able to see that these antibodies can bind the virus, but also can avoid the virus to get into the cells. If there is any specific titer that is particularly protective, we don't know that yet. If there's need of high levels or low level, that is not very well understood, but we know that in some individuals, neutralizing antibodies are not detected. We know for other studies or vaccine candidates that have released their data that certain levels of neutralizing antibodies, particularly, definitely, sorry, I'm going to mention this more in detail. So how do we know that actually neutralizing antibodies are the ones that protect the individual? Because there's different markers. We have the T cell markers, we have other B cell markers, we have binding antibodies. What makes neutralizing antibodies the clue or the key for to induce protection? This assumption was made on prior studies, animal studies that were done with SARS coronavirus in 2003. And additionally, more recently, Harvard Medical School has shown this study that was published, I think that during the second week of May, that shows that rhesus macaques are protected against reinfection, and this protection correlates extremely well with neutralizing antibodies. So the University of Oxford released some data, and this shows that they are able to induce neutralizing antibodies, but if these neutralizing antibodies, certain level is just able to detect against lower respiratory tract infection, like pneumonia or severe disease, or if these neutralizing antibodies will be able to protect the upper respiratory tract too. So that is one of the main concerns about these vaccine candidates, that shows that certain levels of neutralizing antibodies are able to induce pneumonia, are able to protect the individual from lower respiratory tract, from pneumonia and potential severe disease or ICU admission, but they are not able to protect against infection in the upper respiratory tract. And on that end, it has different repercussions in terms of possibility of transmission and herd immunity. Oh, sorry. On the other end, we have the different markers in terms of the T cell activation. We can measure lymphocytes, T cell assays, proliferation assays. One of the main effectors of the T cell response is interferon gamma, and overall, the objective is to identify and if there are SARS coronavirus specific memory T cells that can be activated and protect from infection. But then what about, I heard with concern different statements about herd immunity, that we're going to wait until herd immunity is in different populations for SARS coronavirus 2 to go away. And this is a dangerous misconception, because first, we know that herd immunity is the threshold of immunity in the population that is necessary to provide indirect protection to the ones that cannot be immunized. What is the threshold of immunity depends on different pathogens. For example, measles. Measles is very contagious, and the threshold to reach herd immunity is about 90–95% that is reached with vaccines. So there's different models in the United States that establish what is the threshold of herd immunity in different areas of United States. And this is particularly the one from Hopkins that states that to reach herd immunity to SARS CoV2, there is a need of about 70% or more of the population that will need to be immune. And if we translate these two numbers, it's about 200 million Americans that will have to get infected before we reach this threshold. So, but let's take a look at some numbers to see what is the seroprevalence in different areas after 2 or 3 months that the virus has been in our communities. For example, worldwide, there is about 2.5 million confirmed cases, and up to this data is up to last couple of weeks ago. The seroprevalence has been 2 to 4%. One hotspot at New York with 8.4 million people, and the death rate of around 17,500 deaths that came up to 1 death in every 500. So with such a high rate of mortality, the seroprevalence was just about 15 or 20%. Or let's take the example of Sweden. Sweden is a country that I'm going to mention the death rate because it was one of the countries from Europe that didn't agree with the quarantine measures or social distancing measures. And they had this proposal of reach herd immunity with no restrictions of social distances. So apparently this was a huge mistake because Sweden, as a European country, has the highest rate of mortality per million people. So overall, just of 360 deaths per million people with a seroprevalence of around 30 to 40%. Overall, this approach of Sweden, it cost a lot of people death. So if we take the example of New York, how much would it be, or how many people need to be infected, or how much mortality will be needed to get that threshold of herd immunity? So it has been estimated that it will need, or it will require 25,000 cases per day of infections, and it will take until 2021 to get herd immunity, and will involve around 500,000 deaths or more. Of course, this is a payment that nobody wants to do. It's a high price to get herd immunity, so it's not a viable option. And we also have to remember that these death rates or these numbers are underreported. So probably they were higher than this. So, and considering these circumstances, so we have to consider vaccines as a tool to help us get into the herd immunity. And there is different vaccines platforms into the pipeline. So we have the vaccine mechanisms is a process that includes a version of the antigen to be delivered into the human body to make them believe that is the natural virus and that induce an immune response. So this version of the antigen could be the whole virus that could be inactivated or weakened. It could be a viral vector that is a virus that is inactive and that is considered to be a type of delivery system that includes the genetic material of the S protein injected or inserted into the genetic material of the vector. We have another antigens, I mean, these platforms that include DNA or RNA of SARS CoV2 or protein-based protein subunits of SARS CoV2. So those are the vaccines that are inducing adaptive immune response, that are a specific response and it has memory. And usually most of the projects that are currently in the pipeline involve vaccine platforms involving protein subunits The second ones are viral vectors, and then the third one are DNA or RNA. But then we have the other arm of vaccines that are trying to emulate or enhance the innate immune response as BCG and poliovirus. But these vaccine platforms has different methodologies, has weaknesses and strengths, and we're going to talk briefly about this. For example, the virus vaccines. Virus vaccines starts with an infectious virus, and then it gets to be inactivated through heat or through formaldehyde, and that is the methodology that polio vaccines or hepatitis A are using. And then we have the other viral vaccines that are attenuated vaccines, virus that starts with an infectious virus, and then it goes through different cell cultures. It passes different times, 1 time, 2 times, 3 times, and then it gets different mutations into genetic material, so it makes them inactive or non-infectious. So under this technology, or until this methodology, we have the MMR vaccines. Overall, there is 9 projects that are using these vaccine platforms in the pipeline for SARS CoV2. Probably the ones that has more notoriety are Sinovac from China and the one from Wuhan Institute of Biologics. Then we have the viral vectors. Viral vectors are viruses that are inactive because they have been created, or they are created through genetic engineering, so they are inactive. The genetic material is not infectious. And then it could be, usually we work with adenovirus and measles too. And then a part of the genome of SARS CoV2 that is of our interest, in this particular case, coding for the protein S, it gets inserted into the genetic material of the viral vector. So here, this is a representation. We have the genetic, the segment of DNA for the S protein that is injected into or inserted into the viral vector DNA. So these viral vectors could replicate as measles, and that is a technology that use Ebola vaccine or dengue, or we have the ones which their genes has been inactivated, so they are not able to replicate as adenovirus. There's about 25 vaccines projects under these platforms of viral vectors, and the most common ones are the ones that has been in the news recently is Cansino, and the one from University of Oxford. Then we have the DNA, RNA vaccines that pretty much they have the genetic, the instructions in little segments of genetic material that could be RNA or DNA. And the only situation or the only consideration is that they come up with a delivery system. RNA needs to go into the cell membranes, and it gets encased into liposoluble nanoparticles. And then for DNA vaccines to enter into the cells, into the human cells, they need an additional process to open the pores of the membranes of these cells. And this can be extrapolated, or usually for practical purposes, there is just like a little electrical impulse that goes after the administration of the intramuscular shot. There's about 35 projects that are using these vaccine platforms. They are not licensed vaccines using DNA or RNA platforms, but they were very close attempts done in 2003 during SARS coronavirus outbreaks, avian flu, swine flu in 2009, and Zika virus in 2016. Currently, the ones that are more advanced into the pipeline for under RNA or DNA technology, we have Moderna with an mRNA vaccine, BioNTech from Germany with mRNA vaccine, and Imperial College of London with innovative self-replicating RNA vaccines. And then we have the protein-based vaccines that pretty much are using, instead of using the whole virus, they're using a particular protein subunits. In this particular case, all the attention goes to the spike protein as a whole, or the RBD, the receptor binding domain. So this is all technologies, traditional technology that works really well for under certain circumstances. There is about 52 projects that are using these platforms, and that is already, there's different vaccines that are used these platforms and that we have used for many years, like the hepatitis B vaccine, HPV, and tetanus. But then each of these platforms has advantages or disadvantages. For example, these ones, the inactivated vaccines or the attenuated vaccines. The advantages are that their traditional methodology, traditional technology that is already in place. Is a straightforward process used for different vaccines. Infrastructure for manufacture and production capacity is already in place. And historically, from prior studies in animal models for SARS coronavirus, historically are linked to high levels of neutralizing antibodies. Then the disadvantages is that they require extensive safety testing because we have to start with a virus that is infectious and it has to grow. It requires huge volumes, around 10,000 or 100,000 liters of reaction material. And one effective dose requires lots of viral particles. Then in terms of the viral vectors, the vectors that are these viruses that are pretty much like delivery systems, we have the advantage that no infection virus needs to be handled because we don't deal with the SARS CoV2 per se. We just use the genetic material of SARS CoV2, a little segment that codes for the protein S. There's excellent preclinical and clinical data for emerging viruses like in the MERS coronavirus outbreak. There's a disadvantage though, that this vector that is used as a transport or as a delivery system usually is a virus that is in the community. And sometimes there is pre-existing immunity against the viral vector. Like for example, Cansino. Cansino is one of the vaccines projects that is using adenovirus 5, that is very well known, that is in the community. So, and there's a problem with a pre-existing vector immunity because this affects negatively the vaccine effectiveness because interfere with the synthesis or the response of neutralizing antibodies. There is usually, historically, these viral vector vaccines from animal studies with SARS CoV2, they are associated with antibody-dependent enhancement. That is, these adverse reactions secondary to the vaccines that produce liver injury or tissue injury in the lung. And usually they are well known because they don't produce high levels of neutralizing antibodies. And that is what it shows for the University of Oxford. The recent results show that the rhesus monkeys, they developed neutralizing antibodies, but they were not very high. And we're going to see that in detail. What about DNA or RNA vaccines? It has the advantage that they're fast. There is a methodology that is safe, easy to develop. They don't require lots of volume. We can have 1 million doses in 1 liter of reaction material. To produce, they need just the genetic material that is created by genetic engineering. They don't need to deal with infectious virus itself, and rapid production is possible. About estimates of dozens, millions of doses in a week. So DNA vaccines has been tested for SARS coronavirus in humans, but it was just phase 1. And the RNA vaccines, they need a specific delivery system, right? That they are encased into a lipid nanoparticle. There is also to be able to penetrate into the membranes of the host. And then there is other innovative technology in which these RNA vaccines can self-amplify. So they require such a lower dose. And then once it's introduced into the human body, it can replicate, so not a large volume is needed, and allows those to require lots of S protein to be expressed, because at the end the RNA goes into the ribosome, and the ribosome is the one that is producing this translation process to produce S protein from the RNA. The disadvantage is that they are unproven. There are no licensed vaccines that are currently used using this technology. And historically, based on prior studies with SARS coronavirus, they were not the best in using high levels of neutralizing antibodies. DNA vaccines particularly, they need a specific delivery device, like they need a particular electric impulse to be given after the intramuscular administration of vaccines to assure that the vaccine or the DNA is going to enter into the cells. And the protein-based vaccines that could be subunits of the SARS CoV2, like the spike protein, the advantages as this platform is that it's not infectious. No infectious virus needs to be handled because at the very beginning, at the very, very early stage, it might need an infectious virus to be processed and then to extract the part of the S protein. But there's different models from different vaccine candidates that are including the recombinant technology to produce this S protein, similar to what happened with hepatitis B. So protein vaccines, or the ones that use protein subunits, they are regularly known because they produce high levels of neutralizing antibodies. It's traditional technology, has been used before with the experience of SARS coronavirus, recombinant S protein. There was prior experience in animal models. They protected monkeys, but they didn't have enough time or enough money to test it in people. The disadvantage is the global production capacity. It might be an issue because it requires, at least during the very first stages, to deal with infectious virus and to be able to isolate the S protein to be used as one of the protein subunits. And the antigen or the epitope integrity needs to be confirmed, because if the antigen is not perfect, it wouldn't produce a specific antibody. That is one of the factors that induce this type of antibody-dependent enhancement. For example, when they tested this type of vaccines for SARS coronavirus in the past, they initially used the S protein as the antigen, and they saw that there was some type of liver injury in animal models. So they decided to modify the antigen and to make it a little bit perfect, in the sense that they tried to get all the other components of glycosylation that apparently will prompt this type of response, and they just use the receptor binding domain. And is the current methodology that is used by Texas Children's, that is using a recombinant protein as their vaccine candidate. But we have to keep in mind that we don't have ever made a vaccine for any coronaviruses. Researchers were very close during SARS, within the SARS outbreak and MERS outbreak. So there was already some work that was in place, but because these outbreaks pretty much disappeared and there was very localized into one particular part of the world, and they ran out of money or funding, so they were not concluded in terms of their trials in humans. So then, but we have to consider if the expectations are reliable, because we have heard conflicted information from biotechs and pharmaceutical companies, from the White Task Force, from SARS coronavirus 2, in regards to that possible vaccines might be available by the fall, maybe October, maybe September, and that just seems very unrealistic, because first, the vaccine development is moving fast, and it has been moving to in a speed that hasn't been seen before. But then, how reliable are the promises that they might be available, some vaccines, to the public for by September or October this year? That is just simply unrealistic, and we're going to understand why. So, and this offers, or this expectation, has been obscured by reports from different vaccines like Moderna and University of Oxford that says that they may have some vaccine available by October. I mean, if they have some vaccines by October, they might be available in a very, very short scale. It might not be available by the whole public. So, and the most important thing is that we are not preparing for a second wave, that it could be in expectation, we're expecting that this vaccine might be available, so we are not preparing for a second wave that could be even worse than the first one. So, but there's some challenges that has to be mentioned. We have to mention that there's new vaccine platforms that might require new plants. They require production capacity that is not available to produce output levels. Additionally, there's more than 141 projects, and many of those projects are done by academic labs, and of course they don't have production capacity. How many doses will be needed by the end of October or September? We have to consider that several of these candidates are including 2 doses per vaccine. So the total amount of doses has to be divided by 2 to see how many people might be able to get immunized if there is any vaccine available by the fall. And then another concern is that countries that are home to vaccines production facilities with production power as China, Indonesia, India, or Brazil. So it's understandable that they may want to meet their domestic needs first before other countries. But there are some facts, and we have to realize that there is the administration, or the government administration, has provided some monetary help to different pharmaceutical labs. For example, there is this authority that is called the BARDA, that is a Biomedical Advanced Research and Developmental Authority, that has spent $500 million and has been divided into 5 pharmaceutical companies. So now, who chose these companies to be held by BARDA? So apparently NIH, they were not involved with the selection of these pharmaceutical labs in terms of decide if they are a good candidate to provide all this help or not. And then there's multiple candidates because we need more than one vaccine. I mean, how many people there is in the planet? Around 8 billion. So 8 billion doses might be needed by next year. So only one vaccine might not be able to fit this need. So, and then if we are thinking about, there is one particular set of patients or individuals that are at high risk that will require the first doses of vaccines. So probably the healthcare workers. How many healthcare workers there is in the world? So these are estimations that were done during the 2009 H1N1 pandemic that estimated that 2% of the global population is composed by healthcare workers, and that will involve 156 million people. And then the other fact that we have to acknowledge is that production capacity from China, India, Indonesia, and Brazil, they might be willing, or they might be willing to want to meet their domestic needs first before meeting the needs of other countries. And the particular concern is that different labs that are based in the United States and they provide vaccines to the United States and Europe, the production capacities or the manufacture work is in China, Indonesia, and Brazil. So there's another concerns. So, but then if we have this, if everything goes well, and if we have enough vaccines or a certain limited number of vaccines, so who should get it first? First are considered to be patients that are, or subjects that are at risk. Usually are considered healthcare workers, patients that are older than 65 years old, and people with chronic conditions. So we have to acknowledge that probably college students and children that go to school, they might not, vaccine might not be available for them. And also probably will be until the end of 2021 that the vaccines might be available for the public. And that is if everything works smoothly, because as we know with testing or serological testing or PCR testing in the United States, there was different struggles. There was no reagents, there were no tools to collect the specimens. That was pretty much unexpected, and something like that may happen in the vaccine development. So a vaccine is too far into the future for us to rely on. And when vaccines gets to be available, so probably the demand is going to be enormous and the supply will be minimal. And this is the analogy of probably the demand and the availability of vaccines could be compared of filling a lake with a garden hose. So pretty much that is going to be the meeting the needs of a vaccine at the very beginning. So, but there's multiple vaccine candidates that are needed because as we know, 90% of the vaccines will fail along the randomized control trials. And for that, we need around 15 or 20 vaccine candidates that go into or starts the randomized control trials to have at the end maybe 2 or 3 vaccines that are available that show safety and efficacy. But it's not possible to predict which strategy will be faster or which strategy will be more successful. So, but we know that we have to prioritize safety and efficacy with an adequate randomized control trial. So the development of the vaccine follows a very labor-intensive, time-intensive, very strict process and oversight in the United States. It starts with an exploratory stage where the antigen, we decide what is the antigen that we want to use. It could be natural, it could be synthetic, it could be the whole virus, it could be one particular part of the protein of this virus, it could be RNA, DNA. Usually the exploratory stage takes about 2 to 4 years, and then it goes to the preclinical stage that includes tissue cultures or cell culture systems. It includes the animal testing with mice and monkeys. The objective is to assess safety mainly and immunogenicity. We can have information about a dose, safe dose, method of administration. Challenge studies are also done during the animal, this preclinical stage. Once the animal is vaccinated, Then after a certain period of time, they're challenged with a virus that is our target. And then based on these results that might last 1 or 2 years to get to, so the antigen might be adapted. And that is what happened, for example, when one of these vaccine platforms that are using recombinant proteins, they, at the very beginning, they used the whole S protein as a vaccine platform, but then they saw that they produce side effects, severe side effects, and then they try to modify this antigen and they just use these, the receptor binding domain. But once the animal model has been obtained, and we have to be sure that the animal model is an animal model that resembles the susceptibility of the human and how human is affected by SARS CoV2, I mean with the same severity. And that is still, we are still very early to know if actually monkeys are the adequate animal models for SARS CoV2 infection in humans. But let's say that we have the adequate animal models, so then we go into the randomized control trials, the phase 1, that includes tests for safety, tests immunogenicity, in usually are healthy individuals between 18 years old and 60 years old. It's open label, they determine tolerability, safety dose. The sample size is between 10 and 100 and might take between 4 to 6 months. In some scenarios, the challenge model can be introduced into a phase 1. So the patient is vaccinated and then the subjects are vaccinated and then are challenged with a virus that could be of a minor strain or a modified strain that it might not be as virulent as the virus that is the target of our study. In phase 2, we continue monitoring safety and immunogenicity, but it includes some numbers of vulnerable individuals that are in the vulnerable groups. For example, patients that are more than 65, sometimes include healthcare workers, and some of these vaccine trials are including these individuals as part of their subjects. They are randomized controlled placebo groups. They try to determine the optimum dosage, delivery method, the schedule, and usually is how many individuals they need, between 100 and 1,000, and usually it takes 1 year or more. If they have their answers to these different questions in phase 2, so they go to the phase 3, that is the clinical efficacy and safety in a large group of people. So they take a heterogeneous population, it's randomized controlled trial, double blind with a placebo group, and the sample size is between 1,000 and 10,000 individuals, and it involves longer times of observation, like several years. And what is the objective? Why does it take that long? Because we need safety signals. We need to see if unusual adverse events are able to emerge during these long follow-ups. For example, frequencies of adverse events that are higher than 1 in 10,000 individuals, so are very, very difficult to emerge in these prior studies with a small amount of people and low and a short-term follow-up. And then once information is obtained in phase 3, the data is evaluated by the FDA. They try to see if the data is compelling for safety and efficacy, and the whole process takes about 10 months. But the whole situation that we have to remember is that unusual adverse reaction, I'm just going to move this here. So this unusual, the sensitivity of detection of uncommon or rare adverse events, or the ones that has an answer that is delayed, is low in this trial. So they need a bunch of people that are followed during a long period of time. And we're going to see example of this. So how many vaccines are being tested now? So there is about, at least until last week, there was about 140 vaccine candidates. 12 candidates are already as randomized controlled trials, and the rest are in preclinical studies. So they're in animal studies. So if we can see what is how advanced, or the 4 that are more advanced into the pipeline. So we have here Cansino, that is in phase 2, that is the adenovirus, the viral vector vaccine. And in phase 1 and 2, we have Moderna, that is the mRNA vaccine, Oxford, that is the chimpanzee adenoviral vector, and Sinovac, that is the attenuated vaccine. All the other ones, as you can see, the great majority are in preclinical stages still. And this is we can see how they are distributed, all these clinical trials along the world. In the United States, we have 4 randomized controlled trials. We have Inovio, Moderna. In Canada, there is a development or the project for this oral vaccine. In Europe, we have BioNTech in Germany that is testing 4 types of mRNA vaccines. And in University of Oxford, we have the chimpanzee adenoviral vaccines. In China, we have 4 randomized controlled trials. The ones that are that has been released data so far are the ones that include Cansino biologics, that is a viral vector vaccine, and Sinovac, that is the inactivated vaccines. Additionally, we have the mRNA nanoparticle in Australia. And these are just few of those, because all the other ones, most of those are still in preclinical stages. And this is a brief description of these vaccine candidates. And I'm just going to mention 2 words about each one. Moderna, that is an mRNA vaccine, is in phase 2 and is the one that is associated to the NIA. Cansino Biologics in China in phase 2, that is the adenoviral 5 viral vector vaccine. They're using the same methodology that they use for the Ebola vaccine. So they have some background work done already. Inovio Pharmaceutical, it's including is a DNA vaccine that are in phase 1, encoding for the S protein. This is a vaccine candidate that requires a tiny electrical impulse to deliver the vaccine and to assure the adequate entrance of the DNA into the cell. Oxford vaccine, that is the chimpanzee adenoviral vector that is associated with AstraZeneca, and additionally, the United States has already bought 300 million doses. They have already some background work on MERS. Sinovac from China, that is using the inactivated version of SARS coronavirus, might need a booster. And BioNTech in Germany, that is using mRNA vaccines, 4 different types, because they have to see which one of these different formulations of mRNA might work. And then University of Pittsburgh, they have very innovative technologies too that we can see in some of these projects, and they developed a prickly patch delivery method that is a patch that has 400 microneedles that are made with sugars, and inside of these microneedles, there is the S protein. So the methodology involves that when this patch is in contact with the skin, so these microneedles will melt, and then the antigen will be incorporated by the antigen presenting cells in the skin, and it will promote the immune response. And then the Imperial College of London has another very interesting methodology that is the self-replicating RNA vaccine, pretty much with a very, very low dose of RNA segments. So they introduce into the body, and the body is able to create multiple copies of that. So the advantage is that require a very low dose to start with. And then, so we have talked about the vaccine platforms that are stimulating the adaptive immune response that is specific, that has memory, but we have the other type of vaccines that are stimulating the innate immunity. And one of these, or the representations, or the representative of this is the BCG. So BCG is a live attenuated vaccines that is part of the immunization protocols at birth of some countries where there's high levels of tuberculosis, and they prevent against serious forms of tuberculosis in children, meningitis, TB meningitis, or disseminated TB. And the link or the relationship between BCG and SARS CoV2 has been always very intriguing, based on the fact that there was different ecological studies during the very, very early stages of the pandemic, where they show the patients or countries that included BCG as a part of their immunization protocols, they have less number of cases secondaries to SARS CoV2, less severe cases, less ICU admissions, and less death. But this was at the very early of the pandemic. Probably by now we have information that probably is a little bit conflicting with this initial data that we obtained. But based on this, there's other randomized controlled trials that are using BCG. And the strategy is that BCG is inducing trained innate immunity. BCG is a strategy that induces trained innate immunity for protection against COVID-19, and pretty much is once these vaccines that have been vaccinated with BCG, and then subsequently they get exposed to the pathogen, they result in the release of cytokines, IL-1 beta, TNF, IL-6, and subsequently, so this produces epigenetic, metabolic reprogramming of the myeloid cells. So here we can see a better representation of this. So an individual is given BCG, and this produces reprogramming of monocytes. And this eventually, when they get stimulated or presented with another antigens, it produces IL-1 beta, TNF of IL-6 with the main objective of pretty much eliminating the viral antigen. So it has been demonstrated that it has produced good effects in RSV infection, influenza, herpes 2, and the whole mechanism is that this representation involves the first contact of the individual with BCG at the first time, and this is the type of response of the innate immune system. A subsequent exposure with the antigen that is our target will induce this type of response that is a trained response. And this is secondary to that the subsequent exposure will induce metabolic reprogramming, epigenetic enzymes, epigenetic changes, and pretty much provoke that the genetic material of these lymphocytes or monocytes will open more, and they promote more an open chromatin that will improve the response during the secondary challenge. At the end, pretty much with the objective of eliminating the agent. It could be a virus or it could be a bacteria. So under this premise, BCG has been postulated that probably could be of use as preventive measure for COVID-19. And the hypothesis is that BCG vaccination is a potent preventive measures against SARS CoV2. And there has been different conflicting data about this because there are some arguments that they were saying, well, if other people, if they got vaccinated when they were young, how long this immunity will last until they are older, because probably they already lost this enhancement of the innate immune response. And the possible explanation for that is that probably the main effectors were children. Children were easily or recently vaccinated with BCG. They were not infected by SARS CoV2 because of this enhanced innate immune response. And subsequently, there was less spread of the virus to the elderly. So there was multiple studies. These studies, randomized controlled trials, are done in people that is considered to be at high risk. Healthcare workers, all the elderly, and there's different randomized controlled trials in the Netherlands, Australia, Greece, United States, France, Uruguay, and pretty much the endpoints of these randomized controlled trials is to see reduction in the incidence of SARS CoV2 infection, reduction of severity of COVID-19, and the effect of BCG on time away from work. But so far, with this information, the only thing that we can recommend is that BCG has just to be used in a randomized controlled trial setting, because we are not sure about the efficacy. And most importantly, we don't want people just to be getting BCG and putting at risk all the supply of BCG for children from areas that has high prevalence of tuberculosis. The other thing is that the unknown efficacy is linked to the ecological studies, because these studies that show the countries where BCG was given as a part of the immunization protocols at birth, they were population studies. They were not individual studies. And there's different bias because populations usually may have different genetic structure, demographic structure. Usually the way that they rely information about incidence or prevalence might be different. And also they might have non-pharmaceutical interventions secondary to SARS-CoV-2 might be different. So we cannot get conclusions or reliable conclusions about ecological studies. Additionally, we know that SARS-CoV-2 is a potent trigger of pro-inflammatory responses of immune dysregulation. If the patient is given BCG and after that is naturally exposed to SARS-CoV-2, we don't know if this subsequent exposure is going to promote further inflammation or adverse reaction. So we don't know that for that reason. Additionally, the duration of children. So what about if children is vaccinated very early when they are few days of life? So then what is the effect of subsequent vaccines into this innate immune response or enhancement of the innate immune response? So nobody knows about this. So based on that, so we just have to rely on BCG in the settings of randomized controlled trial. But there's different approaches that we can take, and there are actually different vaccine candidates are taking to save time in vaccine development. The main objective is to save time around the logistics of the randomized controlled trials phase 3, but no compromising safety or efficacy. So now the vaccine development is a process that usually takes under regular circumstances between 20 or 26 years. If you remember, rotavirus vaccine, the pentavalent live virus vaccine, usually took 26 years before being licensed. So initially, overall, in general, it needs a proof of concept with an adequate animal model. So we have the dose of the antigen that we want to use, and if we are going to administer this to our animal model, and it's going to produce a subsequent immune response. So then we have phase 1 and 2 with subsequent larger number of people, and then we get to phase 3 with prospective placebo-controlled trials, and it involves lots of people and it involves long follow-ups. For example, for Rotatec, we have 70,000 person trials that was followed over 3 or 4 years. Rotarix, 60,000 person trials. Pneumococcal conjugated vaccine, 30,000 person trials. HPV, 35,000 person trials before getting the FDA license. Something similar we are expecting to get, but pretty much skipping major steps. For example, to skip animal models or to skip the ideal animal model, because so far we have seen different reports that are using monkeys, that are using mice, but if they are adequate, if we are probably it's too early to determine if one particular animal model is the ideal to imitate or to mirror human infection secondary to SARS CoV2. Then is followed by trials in which the trial that will use a dose, and we will assume that that specific dose will induce an immune response that is protective to a small number of people, and then we'll go into the phase 3 with placebo prospective controlled trials that would involve maybe 1,000, 3,000, 6,000 people. With those numbers, it's uncertain if the vaccine will be effective, because probably sometimes we need even 10,000s of people, several thousands, to be confident that the vaccine is not going to produce an unusual adverse event. So with those numbers, probably it's not enough to tell if any uncommon adverse effect from the vaccine might be available. And obviously, during this, with this new timeline, the FDA license that takes about a year so far might not be needed, or they are planning to skip that part. And that is what happened in the past, in an emergent situation when a lot of people was dying in West Africa. So that is how the Ebola vaccine was given into people. It was released without FDA licensure. It was given to thousands of people, and then it was shown to be effective and safe, and then it was licensed. Other measures to shorten this process is manufacturing risk. At the same time that the randomized controlled trials are taking place, simultaneously manufacturer capacity has been created. And this is a risk, because at the end, these different vaccine candidates are creating manufacture capacity without knowing if their vaccines might be safe or might be effective. And at the end, if they happen to notice that they are not safe and they are not effective, so probably they will just toss it. But it's a risk, and the government or the administration is helping to mitigate that risk. As BARDA, with this a huge amount of money that has been distributed between different pharmaceutical labs. And then we have the very well publicized challenge testing that is a way to shorten this process. So normally in vaccine studies, once they have the vaccine in phase 3, they're usually given to subjects, and placebo is given to the other group, the control group. And then we have to wait until they get naturally exposed to the virus. But what happens if there's no any more virus in the environment? What will happen if the outbreak is already gone? So this means that the experiment will require more people and require more time. And such testing, there is a different alternative to approach this with controlled human infection, in which the individuals or the subjects are exposed to the virus in a lab after receiving the vaccine candidate to find out about effectiveness. So there is a profile of reasonable risk benefit. For example, There is the pros. The coronavirus might be a relative good candidate for challenge testing because young people die so rarely from this disease. And over again, so well, challenge testing is a very well standardized process. It's a long, very time consuming process. Just the protocol might take 1 or 2 years to be determined because they need to know what is the dose, what is the route of administration that mirrors natural exposure to the virus. So probably it's easier or faster to finish with the phase 3 trials instead of waiting for a challenging testing. But there has been lots of publicity about this and the ethical reasons, considering that what happens if these people get infected with a virus and they have severity secondary to this subsequent infection with a virus when there is no treatment or approved treatment, specific treatment for SARS CoV2. So the pro arguments for the controlled human infection is that first, the animal model, we're not even really sure if the animal model secondary to rhesus monkeys is the ideal that will imitate natural exposure from a human to SARS CoV2. What happens if the outbreak is gone? So those are situations that we have to keep in mind to at least to have the plan for these pharmaceutical companies to have that in as a part of the plan, because it takes time. It takes about 2 years to get the adequate protocol to the controlled human infection. If it happens that the phase 3 trials didn't have enough patients or there was not enough circulating virus in the environment, they might take in consideration that approach. And despite being very controversial, there's lots of supports of people around the world. And this is up to last week. There was around 25,000 people that were showing their support, and they were registering to be participants of these challenge trials. Up to now, it's around 27,000 from around 100 countries. But it's still very controversial. But at the end, we have to think that the vaccine decisions and safety issues will be a matter of risk-benefit evaluation, because if at the end we have a vaccine by the end of the year, we have to accept that probably some of the first individuals that are going to get vaccinated are going to be healthcare workers or essential workers. And the vaccine and these are going to be subjected to vaccine administration with no large safety and efficacy trial. But then there is a different circumstances when there is a situation of emergency, of the urgency, because that might make the population or the public to make a different assessment of the risk. So the strategy to adopt, it will depend if there is fear of the virus, because people might be willing to accept some level of risk. For example, if when the vaccine is available, the death secondary to SARS CoV2 is around 1,000 or 2,000 people per day, so probably the population might be willing to accept some level of risk. But if death secondary to SARS CoV2 is around 10 people per day, so the population might not, or the people might decide not to take such risk. But then safety. Safety is a critical goal when providing a vaccine to an individual that is healthy. And based on safety are issues associated with this that involve enhanced disease. And we are talking about the process that when a subject is given a vaccine, that is a vaccine candidate, and then waiting to have natural exposure secondary to the virus, so this subsequent infection might induce an adverse reaction that it will induce tissue damage. So there's historically several examples of this, and one of those is vaccines that were given for RSV in 1960s, and usually associated with whole inactivated formulated vaccines in aluminum. And also there's another virus that mirrors this type of enhanced disease that is a coronavirus. So RSV is one, but RSV is not a coronavirus. A coronavirus in cats shows this type of reaction when a certain vaccine candidates are given. So this enhanced disease that pretty much involves tissue damage or severe inflammatory reaction involves two different syndromes that are associated with vaccine-enhanced disease after exposure to the target virus. One is called the antibody-dependent enhancement. In these circumstances, or during this process, the antigen, that could be an antigen that is not perfect, with low specificity, with several, let's say there is an antigen that is not perfect. They will induce an antibody that is defective. And this antibody will have a particular area in the tail that is the Fc region. This will activate some Fc receptor in different cells, will induce the pathogen to enter into the cells, and will promote infections inside the cells, similar to what happened to one of the vaccines for dengue that induced this type of reaction that is called antibody-dependent enhancement, because it depends on antibodies that are defective. And there's different reasons for which an antibody is effective. It could be because the antigen is not perfect, because the epitopes are defective. And then we have the other type of injury secondary to subsequent exposure to the virus after a vaccine has been administered, that is called the vaccine-associated enhanced respiratory disease, that could be linked to immune complexes. These antibodies are binding, but they are not neutralizing, so the virus can still replicate. And when we have a large amount of virus and antibodies are just binding, so we have these immune complexes that precipitate and produce this type of infection with immune complexes and severe reactions. And then we have the types that are mediated to cell response. And from this, probably the most notorious one that has been historically known is a reaction to the RSV vaccine in the 1960s. So this was a type of T-cell mediated that is called vaccine-enhanced respiratory disease. That type of vaccine was a whole inactivated RSV vaccine, and then was given to infants, and then the infants were exposed to RSV. Well, naturally exposed. So this caused a severe allergic reaction with different cytokines that were released, IL-4, IL-5, IL-13, produce increased mucus production, eosinophil recruitment, airways hyperresponsiveness. So overall, markers of the type of a response that we don't want, that is the Th2, and some markers of Th17. So overall, so we know, and there is some mild agreement into the sense that probably in an adequate vaccine, we want a type of response, cellular response at Th1. We don't want the ones that produce tissue damage or severe inflammatory reaction as Th2 or Th17. As these, the findings is that pretty much this cohort of infants that were vaccinated, 80% require hospitalizations, and 5 compared to 5 placebo recipients, and 2 of those died. And then we see here these tissues with lots of polymorphonuclear cells with eosinophils into the bronchial areas. And finally, so there was 2 deaths. So it was like really a catastrophic event for the world of vaccine development. And this is, well, the hyperinflation of an RSV infected individual. So, and then what is the best animal model for SARS CoV-2? So we have to start with the ideal model. And the situation is that it's still very early to identify what is the ideal model. Is it the rats, or the monkeys, or the ferrets? So what we know so far is that, for example, mice and rats, so they just, when they get infected secondary to SARS CoV-2, so they only get mildly infected. The same happened with rhesus monkeys. So rhesus monkeys are a very promising animal model for respiratory infections, but particularly for SARS CoV-2, they do not mirror the type of susceptibility the humans had for SARS CoV-2, particularly because they do not develop severe disease secondary to SARS CoV-2. So lots of controversies are conflicting data secondary to non-human primates. And then overall, the adequate of ideal subjects for animal models are considered to be so far ferrets or hamsters. And so now, with all the information that I provided to you before, we review natural protective immunity, different arms, different type of response, how long does it take for the vaccines, and for the vaccines development, and what are issues associated with safety. So now you can be the judge based on the claims of these additional 4 vaccine candidates that released data, and they were in the news. So you can decide if they are worth, if they are promising results, because all of those are saying that their vaccines are safe, they are immunogenic, and they are very well tolerated. And so overall, I don't like animal research any more than probably you do, but unfortunately that is the way the vaccine development is done. So one of these studies is the Oxford University. The Oxford University is using the chimpanzee adenoviral vector. So you can see here the viral vector, the adenoviral vector. And here is the segment that is coding for the S protein that is inserted into the genetic material of this adenovirus. So it's a chimpanzee adenovirus. So issues with protective immunity secondary to this adenovirus, so probably it's not going to be that issue. So it's one dose intramuscular that was given, and this segment that is inserted into this viral vector is coding for the S protein. AstraZeneca is affiliated, and the US government has ordered around 300 million doses of this vaccine. So this data that was released into the literature involves animal models with rhesus macaques, and then is measuring neutralizing antibodies and cellular response 14 days after the vaccine was given. And we can see in red are the monkeys, the neutralizing antibodies from monkeys that were vaccinated with vaccine candidates, and in blue were the control group. And we can see that the group that was vaccinated developed neutralizing antibodies that were in a range between 5 and 40. Then these monkeys, after they measured this humoral and cellular response, they were challenged with a high dose of SARS CoV2. And then 28 days after the vaccine. And this is what they found. In the group of monkeys that were vaccinated, they tried to evaluate the presence of RNA of SARS-CoV-2 in BAL, and they detected 2 of these monkeys out of 6. They had viral RNA that was detected. That infers or assume that that this vaccine produced partial protection. There is one particular concept that I want to provide you that is a subgenomic RNA. Subgenomic RNAs are surrogate markers of virus that is actively replicating. So in the BAL of the vaccinated monkey, they were not able to detect, but interestingly, the nose detected as much virus as was in the control group. So pretty much was not protective in terms of infection in the upper respiratory tract. Cellular response were present. Monkeys that were vaccinated were still presenting symptoms of tachypnea. Obviously, the control group has everything that was worse clinically, didn't have any T-cell response. But then the most interesting part happened when they analyzed the tissue post-euthanasia 7 days after the challenge. The lung tissue of these monkeys didn't show any pneumonia. There was no immune-enhanced disease, so pretty much the type of cellular response focused in the lungs was Th1, so that is the one that we are expecting, and there wasn't virus detected in the tissue. So viral RNA was detected, but in lower level, and subgenomic RNA, that is an RNA that gives us the idea that this virus is actively infectious or actively replicating, was 1 in 6. Viruses that were not vaccinated developed viral interstitial pneumonia with high levels of viral load. So you can say, is this information enough? So what happened between the Th1 response? So it was promising in the sense that it looks that probably was just a Th1 response that was generated. But the other, there was some, the question is that probably they didn't show Th2 response because maybe the monkeys were not an ideal animal model. Anyway, they don't get infected, they don't get severe disease secondary to SARS-CoV-2. So that was, they only developed mild disease. So that is one of the questions secondary to this data. So observation based on this data, the monkeys with modest levels of neutralizing antibodies, they were only between 5 and 40. Usually high levels of neutralizing antibodies are considered to be above 100. So between 5 and 40, and with a 40-fold dilution, the neutralizing activity was done, was eliminated, it was not. Usually, very protective vaccines are able to induce neutralizing antibodies in titers of 1,000, right? And these with 1,000-fold dilutions, so they are usually able to stop the neutralizing activity. These neutralizing antibodies, they stop the neutralizing activity only with 40-fold dilutions. So another observation is that all monkeys were infected. So the virus was detected in the upper respiratory tract. So it will infer that probably we extrapolate this to humans, is that will not contribute to herd immunity because this virus could still be able to be transmitted from the upper respiratory tract into other individuals while speaking or while coughing. But the good thing is that this vaccine was able to protect against lower respiratory disease, pneumonia, against disease, but not against infection or transmission. So one of the promising results is that this vaccine candidate was able to prevent damage to the lung and replication in the lungs and decrease severity. So another observation is that this vaccine probably show us that sterilizing immunity probably was not a possibility because these monkeys were still able to isolate virus in the upper respiratory tract. There was one question if there was one observation that probably, this viral load that was detected in the upper respiratory tract. So what about if it was an effect of the high inoculation dose? Because these monkeys were inoculated with a high amount of viruses, and probably the limited amount of the neutralizing antibodies was limiting further replication of this virus, because it's true, we know by this data that there was RNA that was detected in the viral in the upper respiratory tract, but they did not do subgenomic RNA testing in these specimens in the upper respiratory tract to let us know if actually this virus was actively replicating. So they didn't mention anything about T cellular response, SARS CoV2 specific T cells. Promising result is that it was an antibody-dependent enhancement that was shown within their data. So overall, they already are in phase 1 and 2 with 1,000 individuals between 8 and 55 years old. So overall, with this Oxford vaccine, we can say that it was just partially effective with poorly production of neutralizing antibodies. So then we have the Sinovac. The Sinovac pharmaceutical lab, they use the inactivated vaccine that is given through intramuscular administration. They use alum as the adjuvant, and they are sponsored by the Chinese government. It's a safety trial, so they use 12 rhesus macaques, and they gave the vaccine in 3 different dates. They 0, 7, and 14, and they gave 2 doses, medium dose and high dose. And then they evaluated, so we can see here neutralizing antibodies since the vaccine was given, and then we can see before the challenge, because this represents when these monkeys were challenged with SARS CoV2. Before this, we can see this is the medium dose, and this is the higher dose, and these are the neutralizing antibody titers. And here is at 21 days, it was higher. But then, so what happened with these monkeys after this vaccine administration? So we have the vaccine, they have 4 monkeys that receive the vaccine at 3 micrograms, the medium dose, another 4 with the high dose and the control group. And after 3 weeks, these 12 monkeys were challenged with SARS CoV2 to the endotracheal administrations. After 7 days, the group of monkeys that was immunized with the medium dose vaccine, it shows only mild infection. The vaccine in the high dose showed no disease, and the control group showed severe interstitial pneumonia. So overall, the best response was in the high dose vaccine with 6 micrograms dose. They looked for the virus in different areas, in the throat, in the lung, in the rectum. They didn't find any viral RNA. With a medium dose of 3 micrograms, they saw that there was some virus detected, but they name it as viral blips, but the infection was overall controlled. When we'll take a look at the neutralizing antibodies levels, so they were high in both groups, with 145 in the high dose group and 400 in the medium dose. They didn't observe major side effects, and most importantly, they didn't see any antibody-dependent enhancement. So then the question is this information enough so we can answer these questions? So we have to mention that monkey probably is not the ideal animal model. So the number of monkeys is small, so there was only 4 in each group. Other observation is that there was some methodology issues or logistic issues about the isolation of the SARS CoV2 strain that they used to challenge these monkeys. Some of the critics about this is that the SARS CoV2 strain that they used to challenge the monkey might not be similar to the ones that are naturally infect humans. And then there was the main concern about this group. You remember that in the niche after the vaccine was given, neutralizing antibodies was triggered in the medium dose and in the higher dose. So we always have a concern when there's low levels of neutralizing antibodies, because if these neutralizing or antibodies are low level or low titers, or they are defective for some reason, subsequent, there's prior experiences, and when under these circumstances, these animal models gets exposed to natural exposure to the target, to the virus, so they develop antibody-dependent enhancement. So usually they need defective antibodies or antibodies that are in a very, very low level. So the good thing is that they were very fearful for this antibody-dependent enhancement in this group with a medium dose of 3 micrograms, but it didn't show. No lung damage in the monkeys with lower levels of antibodies. Overall, it was considered to be safe at several doses, no adverse reaction, and it showed to be protective. So low-income countries could access this type of vaccine because it's an old technology, it's traditional technology that is inactivated virus vaccine. Overall, we can say about the Sinovac biotech, promising results. They protect all their monkeys, good levels of neutralizing antibodies with no antibody-dependent enhancement. So far, University of Oxford probably didn't look very promising in terms of what type of protection was providing, because several of their monkeys got infected and got virals detected in the upper respiratory tract. But we compare to Sinovac with this type of vaccine platforms, so the results are at least very promising. And then we have Moderna. So Moderna has a phase 1 study, and the vaccine candidate is an mRNA vaccine. It's a safety trial and dosing to identify what is the dose that they want to try. So there's several issues with Moderna that I'm going to mention about this. Unfortunately, there's lots of propaganda, lots of marketing about Moderna, but they didn't release any data. So it's very difficult to have a clinical or critical evaluation of what their claims are. But at least this is a very limited information that they released. They tested 45 subjects in the phase 1 between 18 and 55 years old. 25, and so these ones they divided in 3 groups with 3 different doses, 25, 100, and 250 micrograms. The first 2 doses have 2 doses, and then the description of their findings goes like this. 25 subjects develop binding antibodies similar to the level that develops in convalescent serum. Individuals or subjects measure neutralizing antibodies to plaque reduction neutralization testing. So 4 of these individuals was in the lower dose. In the medium dose, there was another 4 subjects that developed neutralizing antibodies. So this level of neutralizing antibodies, or the titers, according to their description, was equivalent to the level seen at convalescent serums or higher. So safety, grade 3, adverse reactions, redness. So overall, this is very limited. And according to all the data that I already mentioned to you, and probably you are focusing on different questions about, so what happened with the titers of neutralizing antibodies? What happened with the cellular response? So all those questions were in my mind too, and many representatives of the scientific community. The problem with Moderna is that they release results to a press release without data. So the results were given by press release, and this creates a false impression of progress. And the worst thing is that may encourage other teams to do the same. So if you consider some of the questions that you may have, is that, so what happened if there were 45, they just said a subject has neutralizing antibodies. So what happened with the other 37 subjects? We don't know. So what is the profile of these patients with neutralizing antibodies? So they were the youngest group, they were the older group, because if it was the youngest group, so then the older group was not protected at all. So, and then what were the titers? So these titers will protect against infection or will protect against severe disease. We don't know. So there was no correlation of immunity that they were mentioned, or how long this, the duration of protections. They didn't mention, they only mentioned limited A patient has neutralizing antibodies, but we don't know about the T-cell response or cytolytic response. So overall, it seems that it's safe at several doses, that is very well tolerated, no serious adverse reactions, no immunogenic data. So, I mean, we cannot say anything about this, if it's promising or not. They're like hiding results. I mean, it's just, I don't even know if they are hiding it on purpose or just to create more marketing and, you know, to protect their stakeholders. So different reasons for this very publicized vaccine candidate not to show the data. And then the last one is Cansino. Cansino is another adenovirus vaccine that is a viral vector from China, associated with the Beijing Institute of Biotechnology. They have safety trial in phase 1. They try immunogenicity with neutralizing antibodies and T-cell response. So they evaluated 108 individuals between 18 and 60 years old, and they evaluated 3 doses. And overall, they said that the side effects that were quite frequent showed as 50% fevers, 40% fatigue, 39% headaches, and muscle pain. And here, so we can see that about 80% of these patients has some degree of adverse reaction. 10% has a grade 3 reaction, and usually is pain in the injection site was present in about 50% of these subjects. So, and then, so they evaluated binding antibodies and neutralizing antibodies. And we can see at day 28 that binding antibodies measured by ELISA were higher with the highest the dose. The higher the dose, higher the levels, 806, 1445. But then if we take a look of the neutralizing antibodies and we see the levels at day 28, the middle dose has 16, the high dose 34, and you can get an idea that, I mean, those levels, they don't seem to be very high. So, is this information enough? Well, we have information about neutralizing antibodies. Is a modest increase in the levels of neutralizing antibodies. The question is, are high enough to produce complete protection or partial protection? There was some issues. Remember that I was telling you that with the viral vectors, sometimes they use viral vectors that are present in the community and that may have existing immunity against the viral vector. So that is what it exactly showed in this study. So some group of individuals with pre-existing antibodies to adenovirus in a higher titers, 1 in 200, they are the ones that interfere with the formation of neutralizing antibodies. And usually patients that are in the community, usually in the older ages, are the ones that has pre-existing immunity. 50% of these patients has 45, 50% of patients has pre-existing immunity against adenovirus, usually is in the higher group, 45 and 60 years old. This group usually had interference with the formation of neutralizing antibodies. And these adenoviral vaccines, instead of producing neutralizing antibodies against SARS CoV2, was formed like a boosting process for the adenoviral immunity. So, and then, so do not, 50% of volunteers with prior immunity to adenovirus 5 do not have adequate response, was worse at older ages. So there was some, the interpretation of those results is probably good safety, no good safety signs, several side effects or adverse reactions. Maybe the high dose, I mean, should be just deleted from the future randomized controlled trials. Do not elicit a strong immune response because of the pre-existing immunity to adenovirus 5. So that is a big consideration, a big disadvantage of this vaccine candidate. Probably under these circumstances, the vaccine will not be appropriate for people that is older than 65 years old. So overall, this Cansino with this adenoviral vector, neutralizing antibodies that were too low to reach even partial protection, not the best candidate for patients more than 65 years old. So as you can see, I mean, from the 4 trials that I showed you, I mean, 2 of those were animal models, but the last 2 were phase 1 trial or randomized controlled trials. So you can see that probably, I mean, it's just very conflicting data in terms of how effective will be a potential vaccines. I mean, the Sinovac is the one that shows the best response, but it was just animal studies. And to extrapolate that from an animal model that we know that is not ideal into a human being, so if the response are gonna be the same, even though it's the only study that so far showed promising results, so we don't know. So let's get, based on this data, so now you can get reasonable vaccine expectations. If it's shown to be effective, so we will have to set our expectations for some degree of protection against just severe disease. So the vaccine might be able to protect against severe disease, pneumonia, ICU admission, and possibly death. Might not protect against a symptomatic infection or against mild symptomatic infection, because these, as you saw, like these viruses, they were still detected in one of the animal models in the upper respiratory tract. What does it represent for herd immunity, considering that it's still going to be able to be transmitted? So there's no sterilizing immunity to be expected from these potential vaccines. Children might not be able to receive the vaccine at first, because first, in the global picture, even though we know about issues with Kawasaki and pediatric multisystemic inflammatory symptoms, overall, the mortality rate is not significant. And we have to be sure that these vaccines are safe and effective before providing them to children. So overall, we don't know what is the level of immunity that is protective from natural immunity and from vaccine-induced immunity. So what we know from all the experience with other coronaviruses is that the response is short-term and is incomplete. Because for seasonal coronaviruses, the immunity usually lasts 2 months. For the other ones that produce ARDS, might be only lasting 2 years. So we need randomized controlled trial for some vaccines, and we have to acknowledge also that for some vaccines, probably we are not going to be able to identify immunological correlates of protection. I can give you an example. For example, the rotavirus vaccine. We have been using rotavirus vaccine for many years, but we don't have immunological correlates of protection. It has been given to 70,000 people, and certainly it was shown that the vaccine was safe and effective with no immunological correlates of protection. And subsequently, it was given to millions of people. So overall, as conclusions, we have to consider that vaccines need to be effective. We have to consider mistakes in the past. If there was issues with the epitope or the antigen in order to create an adequate antigen to promote an adequate immune response. Safety issues could cause massive delays. So safety issues are extremely important. If we forget about those, and that happened during the vaccine administration, that could not only represent catastrophic events for the subjects itself, but also for the vaccine development in the future. Vaccine candidates and authorities need to be transparent when communicating results of expectations, because big decisions, personal economic decisions, are based on the hope of the vaccines. And then this term that is used by the government administration, Operation Warp Speed, creates unnecessary confusion, because they meant that it means to speed the development of all the manufacture or production capacity along these randomized control trials without compromising safety and efficacy. So that is the end of the presentation. And if you have any further questions, so we can proceed with those.
Thank you. Well, what an immense amount of information. And thank you for compiling all that for us. Regarding these, I don't know if the right word is method or race towards a vaccine or a potential vaccine, is there a threshold where, I don't know if it would be the FDA would say this project, this one company or this one project needs to stop because it's not safe, it's not efficient?
Yes, the situation is that, as we know, and I mentioned during the talk, is that the government administration has been provided different, like a big amount of money to 5 companies, pharmaceutical companies or biotech, for vaccine development. So how they choose which vaccines candidates they were supposed to help, so it's not very clear at all. Because all the representatives from the NIH and some representatives also from the FDA, they didn't have anything to do with that decision. So they don't know exactly how they choose those 5 companies and who is giving this oversight and scrutiny about when these vaccines should be stopped, at what point of side effects should be stopped. I have the feeling that it's the FDA, because they are the ones that are giving all this umbrella of scrutiny and vigilance and these surveillance systems, is not really known exactly. And at the end, sorry to just go into the other situation. So if we know exactly, let's say we have 3 of these vaccines that get available at the end of these randomized controlled trials. One seems to be better than the other one in different aspects. So how we are going to decide, or how these institutions are going to decide which one of those vaccine candidates gets into the population, who decides that? So we are thinking that probably is going to be the NIH, but I'm not 100% sure.
I see. But you just stole my second question. that actually was what criteria, or what was the regulations to determine which one of these ones, that some are more promising than others, even though we cannot actually ascertain they are safe enough and the efficacy is good enough, but how that decision is going to be taken. As you just mentioned, that part of the plan is not clear yet.
Yes, I mean, there is different representatives from the NIH that are, they have, they participate in these meetings, and different doctors that pretty much participate in these huge protocols, multi-center protocols from vaccines for children, and they estimate that probably the decision might need to be taken by the NIH. So now what are the lines or the lineage of requirements that they're going to meet, or they're going to focus on to give the approval for one vaccine versus another one? So that is something that we don't really know yet. But I mean, we suppose that probably is going to be the NIH who takes that decision, and all the representatives that participate here.
I see. And at this point, really, there's none which has, say, satisfied what the NIH would be expecting. Am I right? Is there like a favorite, a runner-up, or something like that?
Well, we know that Dr. Fauci talks a lot about the findings of Moderna, and I'm sure that probably he might have different access to information that probably just the public or the scientific community doesn't have access to. But as I show you, the information that was released in this press conference, I mean, it's just very, very limited. So it's very limited data. They claim multiple things, but as you say, we don't know levels of neutralizing antibodies. We don't know the cellular immunity secondary to that. We don't know the distribution of ages. We don't know many things, but I'm sure that, I mean, Dr. Fauci might have access to all this data, because he seems very, at least mentioned that it's a promising alternative. But I mean,
leaning towards it.
At least from the vaccine platforms that are in the United States. So now, at least the favorite, based on the information that has been released to the public, I mean, my favorite is the ones that presents, I mean, all protocol, all technology that is already in place, that is from China. But now, probably we are not, as a country, we're not going to have access to that vaccine.
Okay. And going back to the first part of your presentation, when you were mentioning herd immunity, and how you would like to see that herd immunity, or the science community would like to think of around 70% of individuals who are considered positive, like the seroprevalence is considered positive. So knowing that for COVID-19, all the tests are not necessarily the best either, that the data is as well confusing, and there's no gold standard for this. In your opinion, what is the impact between those tests not being able to actually capture the true levels of seroprevalence and the expectation for a potential herd immunity?
Yes. So overall, when this very conflicting data was obtained from these serological platforms, so it was established from the very beginning that probably it will make just provide information to take actions on just in the population level, not in the individual level. So having said this, so the antibodies is not let's say, it's not the most accurate information that we get if we're going to make decisions in terms of if this patient is going to be, if this particular subject is going to be at risk or not, if this patient can be re-exposed to a situation that is considered to be at risk. So to make those decisions and to decide if a person is protected or not at an individual level, it has been very established since the very beginning that it's not useful. But it has been seen different uses for the use of these serological platforms at a population level. So to know about seroprevalence, because I suppose, I mean, there's different platforms, right, that they're using in different cities. So different serological platforms are using Abbott, some are using the NIH, some are using, the CDC ones. There's different companies. And so we have a mixture of those, the good ones and the bad ones, and the bad ones have been slowly being released or kind of taken out from the market, as you can see in the FDA. There's different FDA communications that take these platforms that are not considered to be accurate enough or to have enough validation data. They have been removed from the list of tests that are circulating for marketing purposes or commercial purposes in the environment. So it has a mix of tests that are good, that are okay, and the ones that are okay, and based on that, they make a decision in terms of if a reliable seroprevalence can be obtained. And you can see if that relates to the population, you know, because I don't know exactly what might be the seroprevalence in Italy, you know, because, I mean, we can consider spots that has been hit in the same way with the same degree of severity than New York. And I don't know, I haven't reviewed the data from Italy to see what was the seroprevalence, but you can then compare what those platforms show you based on how many cases you had. So, but it's completely understandable. But overall, for population or population studies, so the seroprevalence, the serological studies is just useful for that particular type of setting. If you are taking that as considered as an individual level, if you are protected or not, no, it's not useful, at least so far with the type of testing that we have.
But we could rely more on them for population,
yes.
at a population level.
Yes.
Okay. Okay. So those were the questions that, as I said, you stole one of my questions. So,
okay.
But,
thank you for your questions. And yes, I'm just gonna share the last slide. If at any point you guys have any particular questions, so you can hit me to my email, mabelgutierrez@yahoo.com. And as you know, I'm very aware of your questions, and I will respond them or reply with an answer as soon as possible. Thank you very much for your time and for joining us today. Bye.
Oh no.